Hello friends, so in this lecture we will cover the biological analysis by non-culture based methods. First is the PCR technique, which is essentially the polymerase chain reaction. It is used to analyze bioaerosol samples, and it allows specific taxa present in a sample to be targeted. Essentially, PCR targets oligonucleotide primers, and the sequence of these primers are homologous to the ends of the genomic region of the target DNA. So, we need prior knowledge of the target microorganism and the target DNA basically, so that we design the primer specific to the genomic region of the target DNA. In general, PCR based approaches are relatively fast and potentially can be highly sensitive and could be less susceptible to the false positives, since we are targeting a specific DNA. As with other applications, and including other applications, PCR based techniques can be used to track the infectious agents and the allergenic materials. Now, at the basic level, PCR involves copying the genome sequence and reproducing millions of copies for further analysis. So, we will use DNA material, genome sequences, and make copies of it, and then use that to and assess if our target microorganisms was there or not. This is how it may work like. The first step basically needs denaturing of the DNA. So the DNA is now in denaturing becomes in a single strand. Now in the second step, the primer will attach to the DNA, the single strand of the DNA. So here the primer basically attaches to the DNA. So this is the annealing around at 68 Celsius. So at around 98, we have the denaturing, breaking of the strand into double strand to the single strand. Then the primer is attaching, annealing to the DNA strand. Now you see 5, 3, 3, 5, 3, 5, 3, 5. Now there will be elongation in the next step. So now the primer moves here and the DNA is elongated, with the result being in the next step we have the DNA itself, and now we have two copies. We started with one copy, we denatured, we broke this double strand to single, the primer got attached and then elongated the strand and made another copy of our DNA. So with each strand we got two strands. So that and then basically now we have two copies. Then so this completes the first cycle. Then these two strands will be denatured and they serve as a template for the next cycle. So in each cycle we are basically increasing it by to the power 2. From 1 we became 2 times. Every time the numbers are doubled. So 2, then 2 will become 4, 4 will become 8, and so on. And many copies can be made within several tens of cycles. Once this is done, the PCR can be visualized by electrophoresis. The RT-PCR is a real-time sensing technique, and this is used for measurements of microbes in an environment. It is more of a modification of PCR. So it does not use electrophoresis but employs a thermocycler coupled to an optical module that will measure the intensity of the reactions using hybridized probes and double-stranded DNA dyes. So maybe it can be done in real time. In PCR techniques, biases may be introduced by the nucleic acid extraction method, and the resultant yield and the detection will depend on the detection of diversity may be dependent on the extraction protocol being used. The extraction method should have high recovery if we have a low biomass bioaerosol samples, because we would need sufficient DNA for this process to take place. That is, sufficient biomass needs to be collected from bioaerosol sampling to produce enough DNA which can then be further amplified. So we need to have a good extraction method. Then we have the qPCR, which is the quantitative polymerase chain reaction. qPCR allows real-time quantification of total microorganisms across domains, which could be bacteria, archaea and fungi. This method is widely applied to quantify the abundance and the expression of taxonomic and functional gene markers. qPCR provides quantitative information, that is, the number of copies of a gene target or a target organism, basically the genome, in a sample. So it can give us quantitative information about how many genomes were there in the sample. Often qPCR has been used in studies to detect bacteria, fungi and virus in several indoor and outdoor air characterization studies. This qPCR in general may allow for a more accurate quantification of the target DNA. This could be as a result of the continuous simultaneous detection and the quantification of the signal that is emitted by the fluorescently labeled amplified product during the PCR reaction. The next method which we will just go over is epifluorescence microscopy. The idea here is to use fluorescence. So we have a light source which could be a xenon arc or mercury vapor lamp. Also LEDs may be possible now. Then we have a excitation filter. So this is basically a filter for the incoming light that will narrow the wavelength of the light and only allow that wavelength that can excite the sample. So only some wavelength is allowed to go through to the objective to the specimen. Then we have a dichroic mirror, which basically is a mirror for two types of lights. One, it reflects the excitation light at this place and makes it go to the sample. Now when the sample is receiving this light, it gets excited and then emits some wavelength. This passes through the objective and reaches the dichroic mirror, and this dichroic mirror will allow the passage of this wavelength emitted by the sample. So this dichroic mirror reflects the light from the source to the sample, but allows the passage of light from the sample to the place where we will detect. So now we have a filter which is an emission filter. It only transmits the wavelength that are emitted from the sample and blocks all the light pass through this filter. And then we have the eyepiece or a camera where we will have a image of the sample, and we can then understand what the sample is about. Next we have fluorescent probes. These are probes are essentially molecules that absorb light of a specific wavelength and emit light generally of a longer wavelength. These molecules are called these fluorophores. So these fluorophores are in combination of a ligand are attaching to the protein of our interest, of the target molecule, and then this will exhibit fluorescence, which can be then observed through fluorescence microscopy. So these probes can be applied to determine specific bacterial groups, genera, or species present in a sample. These are examples of some of such probes. 4,6-diamino-2-phenyl-lindole DAPI, which binds to DNA and stains both viable and dead cells. And this can help us count total bacteria and fungi, including viable as well as dead. And we have SVIBR's green, gold, and safe dyes, which bind with double or single stranded DNA or RNA. 5-cyano-2,3-dietolyl tetrazolium chloride, which targets living and respiring cell. Acridine orange, which could bind to both DNA and RNA. And these may be together used to differentiate between cells viable and non-viable. So all these, and then we have some commercial strains. So using a combination of these, one can possibly differentiate between also living and non-viable cells. The next technique is fluorescence in situ hybridization, which is also termed as FISH. It is a technique used to detect and locate specific DNA on the chromosome. For FISH analysis, probes can be designed at different taxonomic levels. Such fluorescent DNA probes are used to target specific chromosomal locations within the nucleus, resulting in a colored signal, and these signals are then detected using a fluorescence microscope. So this is a conceptual figure of the FISH method, where we have the DNA probe and a target sequence. The DNA probe is labeled before hybridization. So this panel, which uses nucleotides that have been modified to contain a fluorophore, these circles. The labeled probe and the target DNA are then denatured. Then these basically then combine. So combining the denatured probe and the target allows the annealing of the complementary DNA sequence, which will then have fluorescence. and then this can be analyzed. The next is dual labeled fluorescent probes. In this, a single stranded oligonucleotide is labeled with two different dyes, a reporter dye which will have some fluorescence and a quencher. The goal of the quencher is to inhibit the natural fluorescence of the quencher. So in the solution there will be some low fluorescence. Now polymerase will elongate. The probe binds to the specific DNA template, and then the hydrolysis will release the reporter from the probe or the target hybrid, and the fluorescence is now much increased. This measured fluorescence will be directly proportional to the amount of target DNA. The dual labeled fluorescence probe method is sensitive, sometimes with reduced possibility of non-specific products. The internal control in each sample can be used to monitor the efficiency of nucleic acid extraction and reverse transcription for RNA targets. The fluorescence generally will increase after each cycle as the amplicons increase. The drawback is that the primers should be fully optimized so that there is no chance of amplifying the non-specific product, because then that would lead to an overestimation of the abundance of target. Because if the primer starts amplifying the non-specific product, that may also give fluorescence, and since our idea is that fluorescence is proportional to the abundance, we are overestimating the abundance. The next method is flow cytometry. Flow cytometry can be used to quantify the structural and componential characteristics of bacteria and fungi. In this technique, the cell is under aqueous phase. The particles are separated in a sheath fluid that is passed through the laser beam. So this is the sheath fluid, and this is being passed through a laser beam. Now in this method, the fluorescence emitted and the light dispersed by each particle are measured with the detectors. So a target specific fluorescence probe is added to the liquid sample, and the cell concentration is measured by light scattering and fluorescence emitted from fluorochromes bound to cell. This is the forward scatter. So this is our beam, this is the cell forward scatter, and these are the side scatters. So there will be forward scattering and side scattering. The forward scattering is proportional to the diameter of the cell. Basically that diffracts light. So forward scattering gives us an estimate of the cell size. Forward scattering. The next is side scattering. Side scattering is due to the light refracted or reflected at the interface between laser and intracellular structure. This gives us information on the internal complexity of the cell. Flow cytometry has the advantage that both physical, for example the cell size, and the chemical traits, this internal complexity of the cell, can be characterized. So it tells us both the physical characteristics, it tells FSC, flow cytometry, SSC and FSC in the flow cytometry will tell us about both the physical and the chemical characteristics of the cell. Then I'll give an overview of microarrays. These are commonly termed as DNA chips or gene chips, which are devices to measure simultaneously the relative concentrations of many different DNA or RNA sequences. These microarrays are made up of a collection of DNA spotted onto a solid matrix, typically a glass slide, for measuring the expression of large number of genes simultaneously, or to genotype the multiple regions of the genome. In general, microarrays may target many more genes as compared to the other methods. Then we have things like microbial volatile organic compounds, which are the product of fungal and bacterial metabolism. These volatile organic compounds need to be analyzed by chemical methods. So the method of analyzing microbial volatile organic compound would involve collecting air sample onto activated carbon medium, and then detection of this sample by gas chromatograph mass spectrometry. MVOCs sometimes cannot be used to quantify fungi, but they can serve as indicators of fungal amplification in the indoor environment. Then we have metagenomics, which aids in the genomic analysis of uncultured biosamples. The procedure consists of isolation of DNA, cloning of DNA into a suitable vector, and transfer to host bacterium. Shotgun is the most commonly used device for metagenomics analysis, and it uses computational power also to generate a clone library. Sometimes these methods not only provide information but also the metabolic processes that may be happening in the community. Finally, we have the gel electrophoresis, which is the separation and analysis of molecules in their fragments. In this method, this method helps us to separate mixtures of DNA, RNA or proteins according to the molecular size. Basically, in this method, the electric field pushes the molecules through a porous gel. Then the molecules will travel through the gel under this electric field, and they will travel at a speed inversely related to their length. That is, the small DNA molecule will travel greater distance through the gel compared to the large DNA molecule, and that will allow us for the separation. So this is the process. In that, first we have the DNA extraction, then we have the isolation and amplification of this, then the isolated and amplified DNA is added to the gel walls somewhere here, and then an electric current is applied. Then we have the size dependent separation of the DNA bands. This is followed by the staining of DNA bands, and then we can characterize the DNA. So in summary, we have looked at various techniques now for bioaerosols characterization. This has ranged from sampling, which was active sampling and also passive sampling, and then also analysis using culture based methods and also non-culture based methods.